Good day, myself is Periyar Selvam, working as an associate professor in the Department of Food Process Engineering, SRM Institute of Science and Technology, Katankulattur. Welcome to the Department of Food Process Engineering YouTube channel. In the current session, I will brief about the importance of redox potential in food for the growth of microorganisms. The factors responsible for the growth of microorganisms are majorly classified into two types. One is internal factor, another one is external factor. In the internal factor, it is going to be covered the pH of the food material, moisture content, water activity or available water of food material, oxidation reduction potential of food material, nutritional type, whether it's a protein food, carbohydrate food, lipid food, according to that the microbial growth is going to be vary, and inhibitory substance present on the food material like antimicrobial substance and barrier property, the structure of the food material. And the external factors, it's going to be storage of temperature, storage of relative humidity, and atmospheric condition or atmospheric gases compositions. It is going to be influence the microbial growth. Apart from that, microbial activity is also going to be covered in the external factors. Why we want to know about the external factors or internal factor responsible for the growth of microorganism means the knowledge is going to be helpful to understanding the food spoilage and as well as it's helpful for the preservation of food material. The OR potential or oxidation reduction potential or redox potential of the substance is the ratio of total oxidizing power to the total reducing power of the substance. Oxidizing means electron accepting and reducing means electron donating. The OR potential of the food determined by four major characteristics. The first one, the OR potential of original food, and second one, poising capacity, that how much it's resistant to change the OR potential from the external source. And third one is the oxygen tension of atmosphere where the food material is storing. And the fourth one is the accessing of external source where the food material is storing. For example, the air having a high tension, whereas the headspace of the canned food having a low oxygen tension. OR potential or oxidation reduction potential written as a EH and OR potential measured and expressed in the term of millivolt, and high oxidizing substrate has a positive EH, whereas high reducing substrate has a negative EH. If the food material is having the high OR potential that favor the growth of aerobic microorganism and also it allow the growth of facultative microorganisms, whereas low OR potential food favor the growth of anaerobic microorganisms and also permit the facultative microorganisms. A fully oxidizing stand oxygen electrode will have an AEH of plus 810 MB at a pH of 7 and temperature of 30 degrees Celsius. At the same conditions, a fully reduced stand hydrogen electrode will have an AEH of minus 420 MB. Usually the EH is going to be depending on the pH of the substance. Normally the EH is going to be measured at the pH of 7. For determining the OR potential or measuring the OR potential of multi-component food, in addition to measuring the each component, we can also consider to check the interface area or microenvironment area to determining the OR potential of the food material. Microbes are classified based on the utilization of free oxygens as aerobic microorganism, anaerobic microorganism, facultative microorganism, and obligate microorganisms. Aerobic microorganism means it's required the oxygen for their growth, and anaerobic microorganism means they are not required the oxygen for their growth. They can be grow in the absence of oxygen. Facultative organism means they can be grow with the presence of oxygen or absence of oxygens. Obligate means it stick to the particular condition. It may be aerobic or anaerobic conditions. They can be grow only in the presence of oxygen means it's a aerobic, obligate aerobic. They can be grow only in the absence of oxygen means that is the obligate anaerobic microorganisms. Aerobic microorganisms required positive EH value or positive MVOR potential. Example of aerobic microorganism: bacilli, micrococci, and pseudomonas, etc. Anaerobic microorganisms required negative EH value or negative MVOR potential. Example: Clostridium and bacteroids. The major group of microorganisms, based on their relationship to EH for their growth, are aerobic microorganisms. Example: Aeromonas hydrophilia. Anaerobic microorganism. Example: Clostridium species, that is a Clostridium botulinum. And facultative aerobes, example Escherichia coli or E. coli, and microaerophilic microorganisms, that is a Campylobacter jejuni. Aerobic microorganisms required OR potential or oxidation reduction potential ranges between plus 500 to plus 300 MV, whereas anaerobic microorganisms required OR potential ranges between plus 100 to less than minus 250 MV. In the case of facultative anaerobics, required OR potential ranges between plus 300 to minus 100 MV. For example, the Clostridium species or Clostridium botulinum is a strict anaerobic microorganism or obligate anaerobic microorganism that required an EH of less than plus 60 MV for their growth, even though slow growth can be occur at the high EH values. The poising capacity of the food is important factor because the poising capacity is nothing but it is not easily allowed to change the over potential of the food material. It's analog to the buffering capacity in the pH. The poising capacity of the food will be affected by oxidizing or reducing constituency present on the food material as well as the presence of active respiratory enzyme system. The fresh fruits and vegetables and mussel foods have low pH value due to the continuous respiration. Most of the fresh plants and animal foods have a low and well-poised OR potential or oxidation reduction potential in their interior part, whereas in the surface and near surface they have a high OR potential and permit the growth of aerobic microorganisms. Hence, in the meat, on the fresh foods, the slime forming or shore forming bacteria growing in the surface of the food material. The plant has the reducing substrate, that is ascorbic acid, and reducing sugars, whereas in animal tissue the reducing components are SH group, that is a sulfhydroxyl group, and other reducing groups are also present in the animal tissues. The interior part of the meat is spoiled by anaerobic microorganisms through the purification process, that is a protein denaturation process. During the processing or heating, the physical structure of the food material is changed and also reducing the over potential of the food component. For example, you take it as a fresh juice and the original pulp. The fresh juice is easily spoiled by yeast compared with the original pulp. Why? Because means during the extraction and filtration process, it's also removing the reducing component present on the pulp. So after removing the reducing component, it easily spoiled by the yeast. In limited amount of oxygen or low oxygen, aerobic and facultative microorganism may produce incomplete oxidizing product. Example, carbohydrate is converted to organic acid. In the case of high amount of oxygen, the product is converted into carbon dioxide and water. In the fructification process, that is a protein denaturation process, occur in the anaerobic condition, it producing the unpleasant odor compared with the aerobic degradations. Redox potential of some foods, example milk, it have the EH of plus 300 to plus 340, cheddar cheese plus 300 to minus 100, Dutch cheese minus 20 to minus 310, Emmental cheese minus 50 to minus 200, butter serum plus 290 to plus 350, egg plus 500, meat, liver and raw minced minus 200, mussels, raw and poster rigor, minus 60 to minus 150, raw and minced plus 225, and minced cooked plus 300, and cooked sausage and canned meat minus 20 to minus 150. Cereals, wheat whole grain plus 320 to 360, wheat germ minus 470, barley ground plus 225, plant juices, grapes plus 409, lemon plus 383, pears plus 436, spinach plus 74, canned foods, if it is in neutral, minus 132 minus 550, if it is in acid condition, minus 410 to minus 550. In conclusion, a food material having high OR potential or oxidation reduction potential, it is possible to spoil by aerobic microorganisms, whereas food material having low OR potential or oxidation reduction potential, it is possible to spoil by anaerobic microorganisms. Based on the OR potential of food materials, we may predict the microbial growth and preservation methods. Thank you for watching. Thank you.